Good morning. My name is Kristen Dana from Rutgers University. Welcome to the workshop, and I will begin our first talk of the morning. Okay, so what I'm going to do is share my screen. So this talk will be about deep learning and computational skin texture. My collaborators are Juan Akula and J&J, and my PhD students, Parneet and Hong. Parneet is now with C Machines Robotics, and Hong is now with Amazon AI. So we've done computational skin texture work for a long time. In particular, we have skin recognition results, and I think good skin classification results using fixed filter methods in the long past. So our papers on skin texture modeling, bidirectional imaging, and modeling of skin texture. These were things that came out before deep learning. But still, some of the basic ideas about using multi-view images of skin are very interesting, and it's not used that often today. So I think I will discuss it a little bit. So this is part of our skin data set. This is several images of the same patient. And you see on the first row and second row, it almost looks like something about the physical properties has changed. This is the corner of the lip. This person had come in to the clinic that we were collaborating with for a laser wrinkle removal procedure. And in this bottom right, it almost looks like, hey, this worked. But the interesting thing is this is the same person at almost the same instant of time. The only thing that's changed here in the individual images is the illumination direction. So this concept of BRDF, how does appearance look as a function of viewing and illumination direction, it is very important when talking about skin recognition. Here's another set of our prior images. This is a basal cell carcinoma. The first row shows the same patient, the same moment of time, almost within a seconds of each other. These images were taken. And each panel, again, represents a different illumination direction. And so that you can see a little better, two of the panels are chosen and zoomed and put right next to each other. And you can see that in the left side, we have a translucency that is, and the underlying structure of the dark patches is visible through the translucent skin. On the right side, though, we see a slightly elevated border. And the dermatologists that we were working with in this project were excited when they saw this because they realized that indeed it's not, neither one of these images gives the full picture. And again, all that's changed is a change in illumination direction. So when you think about applications like teledermatology, and you think about what is the right digital representation to send, this idea of multi-view images is interesting. Here's another example of bidirectional imaging of skin with the skin condition psoriasis. So the texture is clearly important in these images. And the texture doesn't follow any simple mathematical model. So we think about what kind of model, what kind of data-driven model is useful in moving forward in texture analysis for skin. And I like the following texture model guidelines. Certainly a model needs to have intra-class invariance to support recognition, right? All examples of the same texture class should give rise to similar values. It should have illumination and geometric invariance. So that's what I was getting at with the multiple illumination directions. You still want to recognize it, or maybe you want to use multiple illumination directions to recognize it. Geometric invariance to support recognition in robust real-world imaging conditions. And these two really are where deep learning shine. By training a model with many examples, you get that invariance. Okay, so other texture model guidelines, we'd like it to do prediction and synthesis. We'd like it to do change detection, right? The model should support um, small changes and quantifying those changes. And fifth, the model should have a physics-based interpretation. Right? So can you support the inference of physical or sometimes biological properties from appearance? So I'm going to talk about a subset of these things today. So we're going to discuss our recent deep learning texture models for synthesis, recognition, and prediction of surface properties. So as a preview, the synthesis, we're going to talk about face texture transfer, style transfer. That's very popular these days. Then we're going to focus on how to do it for face texture. Recognition, I think the fixed filter methods worked well in the past, but with large data sets, clearly that's not sufficient. The recognition that I'm going to show today, though, is deep learning methods for skin recognition in microscopy images, so that you can recognize what part of the skin you are looking at in a particular microscopic, in a particular optical section, and then do skin depth estimation. And then the final part of the talk will be about predicting surface properties. The prediction is the prediction of microbiome. Okay, and this is a very exciting area, I think. Can you link appearance with microbiome with these sophisticated deep learning texture models? Okay, so first we'll talk about style transfer. So style transfer has received a lot of attention in the literature. We're certainly one of many in the game. But the idea is that if you have an image and a style, an artistic style, you can automatically regenerate that image with that style, right? So here's some examples, the images above and the styles in this first column here. And then each of these 4 images is this image with this style, this content image with this style in the second row, this image with this style, and this image with this style. Okay. And this was the work multi-style generative network for real-time transfer that appeared in the ECCV workshop in 2018. The basic idea is that somewhere in the deep network, you have statistics that capture the style. And if you force a match between the statistics of the original image and the statistics of the style image, and that forced match is done by creating these networks and links in between the different levels of the networks of the style image, of the descriptive network and the transformation network, establish these links and change the image until the output image has this match in just the right places. So let's look at some of the results. Here's an input in the first column. The second column shows input 2. The style, we're going, in this third column, we're going to take the style from the first image, the content from the second image. So we're looking at regenerating this person, but with this person's texture, and that's the result. And we'll notice one of the nice features, and some of the algorithms that we compared it to didn't have this property, that the identity is preserved. This person looks more like this guy with the first person's texture. Okay, and then this last panel, we flipped it. So we have the style from the second image, but the person is from the first image. And you can see the result here. So these are fun to do. Here's another example, the content image, the style image, and the content image with the style image. Another example, content image in the first column, style image in the second, and the third column, it should be this content with the style. And you see the beard, some of the facial texture in the beard comes through. Okay, so the next this is kind of a whirlwind tour. I'm not going through all the paper details of how we do it. I'm going to move on to our recognition work. So we're doing deep learning for classification, but this time not for surfaces, but rather microscopic skin images. Okay, so this is the work from Parneet and myself and my collaborator Wanakula and Catherine Mack. It's called Hybrid Deep Learning for Reflectance Confocal Microscopy in Skin Images. So in an RCM stack, we have multiple skin layers, starting from the outside epidermis, continuing to the stratum corneum, continuing down to the portions of the papillary dermis. And if one can recognize the images in that stack, right, we can determine the thickness of the skin automatically. And that was the idea here. So there's a lot of intraclass variation. And so this, they are labeled by a human expert, but this gives you the kind of variation that you might see. These are all the same class in terms of the layers, but their structure is very different. So it's for recognition, it's very, very challenging, and none of the fixed filter methods would work. But with enough patches in the training images and using a pre-trained CNN, we were actually able to do this at a neural networks classifier. We trained a patch classifier. So that's kind of a trick to how to get more labeled images. You use many, many patches. And of course, we're using a pre-trained CNN, so that helps. And then we do patch classification with the random patches, and then the image classification based on the distribution of the patch labels, and that's it. Okay, so the feature vector in the we take the RCM image, and we also compare a hybrid deep learning. So in this way, we use a texton-based feature vectors that are obtained with pre-built texton libraries, 5x5 patches, okay? And then you do a fixed filtering, texton labeling with max8 pooling, and a histogram followed by a deep neural network. So in this sense, we're doing the deep neural network, but only on this 50 × 1 histogram to get a class label. And it turns out that this works a little bit better than the patch CNN. So these methods are described in our paper. And again, the takeaway is the results are the ability to obtain skin thickness using an RCM stack. Okay, so now the final part of my presentation is answering the question, can skin appearance predict microbiome? And I really think this is a very fascinating open area that we're really just beginning to scratch the surface on. So one of the inspiring papers was Fitzgibbon et al. And the idea in this paper was if you looked at a set of images and divided them into the very simple labels, healthy skin and acne skin, right? So it's sort of a good, bad label. And then looked at the microbiome of different strains on the skin, you might think, well, there should be more bacteria in the bad skin. And it turns out that in the good skin, there was more of certain bacteria, indicating that, and this is not my field, but the general indication is that the microbiome, there's good bacteria that leads to good skin. This is something, at least at a high level, that seems to be well known. So I began to think about what our computational skin modeling can do for this type of work, where you're trying to make a correlation between skin appearance and the skin microbiome. And certainly we can um, computationally characterize appearance in a much more sophisticated way than the binary label good, bad, healthy skin versus acne skin or other diseases or issues. So I began to think about, can we create an appearance model and then use microbiome measurements from people that we have also done extensive photography measurements with, and build a model that links the photographic appearance to microbiome. And we have some initial results that are promising, but certainly as a field that's very interesting, right? It's much harder to measure the microbiome than it is to take a picture. But if you could take a very detailed imaging, perhaps multi-view, perhaps multimodal, um, and relate that in a powerful way with the tools of computational texture modeling that we have, perhaps we could predict microbiome from photography with some confidence. Okay, so here we have UV, blue fluorescence, UV, and polarized skin images. Okay, we did a study in collaboration with J&J of skin images and multimodal skin images and microbiome with 40 subjects. Here's some examples. This is the blue fluorescence modality of the subjects in the study. Here are some examples of the patches. And these patches I chose on purpose because they show what we found to be useful attributes. So one of the things that we found is that if you used some supervision, label these patches with the classes, we can train a neural network in a much better way to label patches as opposed to large regions. We went back and forth on trying to label faces, thinking about whether we should label faces or patches, and supervised patches really helped a lot. So what are the classes? So these are in A and B, we have what we would call blotchiness, G and H as well. In C and I, we see fine hairs, and in many of the in these other images, we see dots, and F would be something called red dots. And we go ahead and give those labels that we manually assigned to patches in the image. So our pipeline, again, is a hybrid of classical text-on-histogram methods and neural nets. We start by using a filter bank over a 5x5 region. We create textons by clustering the output of the filter bank. Again, this is very fixed filter classical vision approach. We use a text-on histogram, a soft-weighted text-on histogram, and then a neural net classifier. And that classifier gives us attribute labels, and then we compute the histogram of attribute labels. And we use that as a skin appearance descriptor. Okay, so that's what we do on the appearance side. On the microbiome side, we have these vectors given to us by the skin microbiome swabs. And these are approximately 700 by 1 vectors. And we just run it through an eigen analysis, and we create an eigenbiome space. On the left, we see all of the subjects in the eigenbiome space. And this has nothing to do with their images, right? So this is just based on their microbiome as measured by the sequenced swabs. And we go ahead and cluster them, a simple clustering. So the red and the blue and the green correspond to their clusters in microbiome space. And we see that indeed there are some clusters, especially this one right here. We noticed this region had a very tight cluster. And just observationally, we said, well, let's see if that tight microbiome cluster, if the subjects in that region, the subjects in that region had similar appearance. And that's what you see to the right. We looked at the appearance for the skin patches for each of those subjects in these red dots. And what you see is this red, I'm using red dot for two things, the red dots to represent the clusters. But then when you look at their images, coincidentally, they are also red dots. This is the attribute for the skin appearance. And this is not really all that surprising because this redness indicates a particular bacteria. So it's just a very clear link between the microbiome and the appearance. So in the paper, we went through and we asked, well, what other links can we make? Where is the conditional probability of a particular microbiome given the appearance high? Conversely, where is the conditional probability of a particular appearance attribute given the microbiome, given membership in the microbiome cluster, where is that high? So we were able to see that for the fluorescence modality, the probability of being in M1, that was that red cluster, given the appearance, is very, very high, right? So the appearance is indicating very, very well what the likely eigenbiome cluster is. Similarly, it was pretty predictive, that 0.85 probability of M2 given the appearance. And then on the flip side, not surprisingly, the probability of the appearance given M1 is high. So we have these indicators that appearance and microbiome are linked, and one can be used to predict the other. Okay, so again, I think this is very interesting start, but it's not conclusive. And actually, the microbiome data is hard to come by. I've been trying to get more of it. So if anybody's interested in collaborating, let me know. And that opens up some interesting possibilities in the skin microbiome analysis. Okay, so here is a list of additional publications from my lab, and that's it. Thank you very much. I hope we'll have time for questions.